Thank you for the introduction, Dr. Yu. That was fabulous. And so are the people in the back hear me okay? Perfect. Okay, thank you all for coming. It's a great pleasure to see all of you here. And today I'm going to talk about my study on exploring plant height plasticity observed in natural field environments. Okay, so for biological research, I think there are two big components: explain and predict. For example, for plant height, no, let's not think about plant height right now, just the plant phenotype. So plant phenotype is a result from genotype, environment, and their interactions. So we usually try to explain plant phenotype with these three components, and we may do an ANOVA to see which component plays an important part for the plant phenotype. And we may also do a variance component analysis to see how much percentage each component is contributing to the final phenotype. And I call that to explain in a quantitative way. And sometimes we also do prediction as well. If you have a plant phenotype and you don't have other information, that's okay. If you discover some patterns among the phenotype, you are also okay to do predictions. And there are also cases when you have the genomic data and you can do genomic predictions. And if you have observed some patterns in the environment and G by E, you can use those patterns to do predictions as well. There's another component for explaining. I call that in a biological way. And this is most focused on the genotype part. We usually trying to explain the genotype by look at the genetics, the cellular basis, or molecular mechanisms, or even in developmental biology aspects. And with this information, that helps us to explain the plant phenotype better, and use those information would help us to do better predictions. There are components for environment and the G by E. Not that much study has been done compared to the genetic side. But these components are important for the final phenotype. And it's especially important for breeding and selection, because if the phenotype keeps changing across different environments, how can you select the best individuals? So we would like to untangle and dissect this problem, and we may explain it. We may use what we found to do better predictions. Okay. So plant plasticity is describing that a genotype behaves differently across different environments. So as shown here, there are two environments, and this is your target trait, and your phenotype has changed across different environments. That's one genotype. If you have multiple genotypes, they are plasticity, and they are all parallel to each other. That's plasticity without G by E. And if you have different variance for the phenotype across different environments, we call that plasticity with variance changing G by E. And also there are sometimes cases there are crossovers, and we call that plasticity with rank changing G by E. And sometimes they are very complicated G by E. We call that plasticity with G by E with complex, continuous, nonlinear functions. Okay. So for the first three types, it's relatively easy to observe the pattern, and then you can use this pattern to make predictions. But how about the last part? So for this type of things, we want to do a pattern discovery to see if there is a pattern to be discovered so we can use to do predictions. Think this of a categorical environment, and I use a dashed line to represent each environment. If we did something to alter this x-axis, maybe sorted, or use a different parameter to represent the environment, we may be able to get a different pattern. That is, some of the environment can cluster together, some in the middle, some separated. Use that, we may be able to develop a still complex but continuous linear functions. So linear is a lot easier than nonlinear to be modeled and then to do predictions. There are different models have been proposed to explain the genetic architecture of G by E. One of them is a QTL sensitivity model that describes that QTL's effect changing across the environment. So in specific, there are four categories. For example, if the QTLs having opposite effect in two environments, we call that antagonistic pleiotropy. And sometimes the effect in one environment is close to zero, and effect in other environment is relatively large. We call that conditional neutrality. And there are also cases we have effects have different sizes in two different environments. We call that differential sensitivity. And there are also cases when there's no G by E. Of course, the effect for those QTL would be equal in those environments. And so this, we would know that QTL respond differently across different environments, and the effect from a major QTL or a combination of multiple QTL can shape the final phenotypes in different environments. Our lab recently has published this work about the plasticity in flowering time in sorghum, and we proposed a joint genomic regression analysis framework to dissect the complex flowering time plasticity observed in natural environments. And we also developed an explicit environment index for prediction. So the goal for this project is to unravel sorghum plant height plasticity using the same framework. And specific objectives were the first one, to uncover the patterns of sorghum plant height plasticity in diverse environments. And second, to predict performance in new environments. And third, identify and dissect the genetic determinants to explain the observed plasticity and G by E interactions. So my experimental design, I have 7 environments, including 2 years in Iowa, 2 years in Kansas, and 3 years in Puerto Rico. So in each environment, I have 2 blocks. In each block, I have 250 recombinant inbred lines. As we can see in the left figure, the population of the plant height is showing in each column, and these 7 environments. They are changing dynamically across environments, and quite a big variation across environments. So first, I did the variance component analysis based on the model that I have. So the ANOVA table showing that environment, genotype, and G by E, all of them are showing significant effect to determine the final phenotype. And next, I look at how much percentage each component is contributing. So environment explains 11% of the total variance, and genotype explains 66%, and G by E explains 10%. So although genotype is still showing the majority of the variance, but environment and G by E together are showing more than 20%. That's still a huge amount for the total variance, so it's worth of study. Okay, so now I want to talk about the pattern discovery journey for this trait. We use a joint regression analysis, and also called a Finley-Wilkinson model. So as I shown you guys here, this is the same graph as before, the variation of the phenotype. And please note the x-axis right now are sorted by geographical location and more specifically on the day length. So if we resort the axis based on environmental mean, that gives us an increasing trend, so relative lower plant height and higher plant height. And then we continue to make the pattern more obvious to replace this categorical axis by numerical axis. And that gives us this figure. So we can see some environments are stretched far away, some environments are clustered together, but the environmental means are showing a linear one in a straight line. And in this case, we would be able to fit regressions on each genotype. And that will give us the final figure. So, and each individual is showing an R square of 0.66. So that means majority of the genotype fit well to the regression line. So from A to D, we have unraveled this complex G by E interactions into simple linear regressions. And with this easier model, it will be a lot easier to do modeling and then to do predictions. So how exactly does prediction happen? So for each genotype, that is one line represented here, we'll have the regression equation for y equals a plus bx. And after you regress, you would have the a and b for each individual differently. And here, the x represents environmental mean, and the y represents plant height. So with that, it gives us, with any given environmental mean, plant height can be predicted with this equation. However, environmental mean is the population mean in that environment. That means that we have to grow this population in that environment to obtain the population mean, and then that's the environment mean. And that works if you want to predict the data you already have, like the past data. But prediction, in some case, we want to predict in the future, and that is more exciting. We can't do that with the environment mean. So how do we solve this? Our lab proposed an environment index to replace environment mean. So this environment index is purely from environmental parameters like collected from weather stations such as day length or temperature. I use JDD here to represent temperature. And we tested 4 parameters. So day length, temperature, and PTT and PTD are combinations of different environmental parameters. All the data can be obtained from the weather station. And so we found that the PTD has the strongest correlation with the environmental mean. And more specifically, if we look at the correlation across the entire growing period from day 0 to day 120 with the different window sizes of like all kinds of different window sizes, we would identify that there's a critical growing point, 59 to 63 days after planting, that has the highest correlation with the environmental mean. That means we only need the PTD data from 59 to 63 after planting to make the prediction. So with that, we would be able to predict without using environmental mean, but use a PTD. So the same here, I plotted the plant height across the PTD, and with each genotype, we can still use y equals a plus bx. And here, x would be environment mean, and y is the plant height. So with any given PTD, we would be able to predict plant height. And as I mentioned, the PTD is combined of certain environmental parameters collected from weather station. So that means we don't have to grow the plant anymore. We can forecast the future. So no more field work, guys. So we want to validate our model. First, we did a cross-validation using Lee-1 environment out. We use if we do the prediction by using PTD, we got a prediction accuracy of 0.88. It's comparable to if we use the environment mean. So that means using PTD, this model works, and it can be as good as using environment mean. And we also want to know if it performs well on the future data. Although this is not really the future, but we do not have this data in our model. So this is an empirical validation test on new environments. So we have 3 additional environments, and the prediction accuracy can be 0.93. So the prediction accuracy is pretty high, although it's off target a little bit. It's not on the diagonal line. That means the predicted plant height is a little bit shorter than the observed plant height. So up to now, I have explained in a quantitative way using ANOVA and a variance component analysis. And we did the prediction by pattern discovery, modeling, and prediction. And what about the explain on the biological side? So I will show some data on the genetics work that we have done to understand better of this G by E. We did the QTL mapping on plant height across all 7 environments, and the 3 QTLs are consistently showing up across all 7 environments. And we plotted their effects across different environments. As we can see, the QHT71 and DW3 are showing dynamic changing of the effects across different environments, and the DW1 is relatively stable. If we compare this effect with the actual phenotype, we can see they are showing the same trend. So the lowest plant height has the smallest effect, and the highest has the highest effect. This shows that the combination of QHT7.1 and DW3 shape the plant height in different environments. And also, as I showed in the introduction of 4 types of different sensitivity model, this one is a differential sensitivity for the QTL effect changing across environment. Okay, so in summary, we have dissected sorghum plant height, plasticity, and G by E by pattern discovery and modeling and prediction. And an environmental index PTD at 59 to 63 days after planting was identified to replace population mean. And high prediction accuracy was achieved using environmental index PTD. We did cross-validation and empirical validation. All the prediction accuracy were high. Differential sensitivity were detected for 2 height QTL across environment, contributing to the observed plasticity and G by E. So with that, we did prediction, and we also did explain. Yeah. So for my future work, I would like to combine some genomic prediction to enable prediction for new genotypes in new environments, and also explore the biological mechanisms and plasticity. With that, I would like to thank all my lab members for contributing very intelligent, useful discussions to stimulate me to complete this project, and especially thanks to my professor, Dr. Yu, and lab mate, Dr. Xiaran Li, and Dr. Tingting Guo, and also Jia Lu for helping me collect a lot of phenotypic data. And a lot of undergrad assistants has helped me a lot. Anna, Angela, and Owen, Andy, Holly has been helping me a lot. And also Greg for helping so much in the field so that I can get all this phenotypic data. And I want to thank my committee members and collaborations, and also thank the funding from Plant Science Institute and our department and the Baker Center. Okay. And thank you all. If you need to contact me for any further discussions, this is my email. Thank you for your attention. I would like to take any questions.
Questions for Chief?
Hey, Aaron.
So your correlation matrix for PTD environmental means,
Yes.
at least from the picture that you showed in my poor color vision, the strength of the correlation looks to be very uniform across all windows. So is there practical variation in the magnitude of the correlation across that matrix? Would you get the same results or very similar ones with different windows?
Okay, so Aaron's asking, because my correlation with the PTT and the environment mean are relatively similar based on the color. So you're probably asking more explanation to that, right? Or yeah. So and first of all, you're right. It's all the windows are relatively high, but it's still having a range from 0 to -1. but a lot of them are relatively high too. But that means that the PTD in the whole is contributing to the plant height. It also relates to the component, this it has to relate to the equation for the PTD, but I cannot reveal what the equation is right now. So let's just say the overall value across the window of PTD is highly correlated with plant height, but there is a maximum, and we can use that window. But it can be relatively flexible for the window to be longer or shorter. Yeah, I hope that, Hey, Cassie.
Wait, so when predicting in new environments, you use your environmental PTD covariates, but you're accounting for, I would agree that you're accounting for most of the variation in an environment by using those weather components, but have you thought about integrating other components of the environment, such as soil and soil water, which obviously has a huge impact? I'm just thinking from a crop modeling perspective, we know that soil and management, those things impact, and if you're trying to predict in new environments.
Yeah. So Cassie is asking if I have considered adding some agronomic related traits as soil or management or rainfall, probably. Yes, that's definitely a future direction. But for now, with the parameter that we collected from weather station, that's good enough. But if we want to further improve the model, that can certainly be done. And if we can combine with the crop modeling, such as epistem, that would be awesome.
So, G, great presentation, by the way. So you talked about plant height specifically in this presentation. Do you suspect that you're going to see as good of prediction accuracy when you move into different traits other than plant height?
Okay, that's a great question. So Dr. Barbie is asking if the prediction accuracy can be as high to other traits. So before I have done this, the paper that we published was on flowering time. And one of the reasons that I did this project is to want to see whether this framework works on different traits. So it turned out for plant height, it's also very high. And of course, the flowering time is really good as well. But I haven't tried to see if it will work for yield. But yield is relatively complex. For a lot of other studies, it didn't work that well either. So, but I'm quite positive that it will work for other traits. Because for different traits, we may be able to identify different environmental parameters that works best for that certain traits. So I think it's possible that, and right now we have 2 traits approved, so I think it should be promising. Yeah. Yes, John.
So you noted the 59 to 63 days or so after planting was the most significant window for predicting. Is there anything, I've never grown sorghum in Iowa, only Kansas. Is there anything going on average in that time period physiologically? Is it hitting a certain vegetative stage at that point, or?
Yeah, that's a great question. So John is asking whether the 59 to 63 is a critical growing period for sorghum development. And the answer is yes. So in my case, 59 to 63 is usually the booting stage. And somehow it's related to the final plant height. But yes, that's a critical growing period in development. Yes, Iron.
So to go back to your discussion of the different types of patterns in the differentials and steady model, those patterns are all predicated on comparisons of 2 environments at a time. And so for your study here, you're using 7 different environments, and generally we know that 2 environments is not sufficient to
Okay, so Aaron is asking whether those 4 categories of QTL sensitivity model would apply to multiple environments rather than just 2. Are they play a critical role in those cases? So I think they are, because for this example only, it's showing differential sensitivity, and the pattern is quite clear in compared to the plant height. And also in our flowering time paper, we not only detect this, but also diagnostic pleiotropy and conditional neutrality as well on different genes. So one of the major flowering time gene, MA6 or MA1, that was showing a diagnostic pleiotropic across different environments. In certain environment, it promotes flowering time, but in certain environment, it delays flowering time. And there are other genes also showing conditional neutrality. And the relatively pattern for those QTLs are similar to the phenotype. So I do think this model explains a lot.
Good answer to an oral prelim question that we just got there.